You know, inside every single one of us, there's a universe, a bustling world of trillions of microorganisms. We call it the microbiome. And for the longest time, it was a complete mystery. But that's all changing. We're finally learning to read its language, to decipher its stories. So today, we're going to explore how scientists are taking this incredibly complex biological data and turning it into a readable book. A book that might just hold the secrets to our health. All right, so here's our roadmap. We're basically going to follow the steps of a data detective cracking a huge case. First, we'll grapple with this chaotic world of data. Then, we'll find the key players, the main suspects. After that, we'll map out their entire community, uncover their group behaviors, and finally, see how all of this can actually predict our health. Ready? Let's go. So first things first, we really need to get our heads around the sheer scale of this problem. I mean, this isn't like looking for a needle in a haystack. It's more like trying to find a specific needle in a haystack the size of a planet.
Seriously, think about this for a second. How would you read a book with 1 trillion letters? That's more letters than there are stars in the Milky Way galaxy, and it's all written in a language you don't speak, with 0 punctuation. That right there is the challenge of digging through the microbiome's genetic data. It is just an insane amount of information.
And the stakes for learning this new language, they couldn't be higher. Every one of us has a totally unique microbiome, this delicate little ecosystem. And when that balance gets thrown off, the technical term is dysbiosis, it's been linked to some of our most serious diseases, things like Crohn's, obesity, even cancer. So reading this book isn't just some fun science project. It's a critical new frontier in medicine.
So where does our data detective even begin? Well, the very first step is to turn all that genetic chaos into an actual list of suspects. We have to figure out who's at the crime scene, and we do that by reading their genetic instruction manuals, which tell us not just who they are, but also what they're capable of doing. To do that, scientists use this incredibly powerful tool called a pangenome. Now, just think of it like the complete dictionary for a bacterial species. It has every single gene that species could possibly have. It's the entire toolbox of functions it can draw from. Then, this really clever method called panflan comes in. It takes the raw DNA from our sample and it compares it line by line against that huge pangenome dictionary. It meticulously checks which of the thousands of possible genes are actually present in the microbes living in this specific sample. And boom! Here's the first huge breakthrough. All of that insane complexity gets boiled down into the simple, clean grid. For every sample and for every gene, you get a straight answer. Is it here or not? A 1 for present, a 0 for absent. Just like that, we've turned a sea of noise into a clear list of suspects. Okay, so we've got our list of who's in the room, but now we need to figure out their relationships. Are they working together? Are they partners in crime? Or are they bitter enemies? The key tool for this is something called correlation. It's really just a statistical way of asking a simple question. When the amount of microbe A goes up, what happens to microbe B? If B goes up too, they're probably friends. That's a positive correlation. If B goes down, they're probably enemies, a negative correlation. And if B doesn't do anything at all, well, they're just strangers. And what's so cool is you can actually see these relationships. It's so clear on these charts. See how the dots form a line going up? That's our friend's relationship. And the line going down? That's our enemies. But if it's just a random cloud of points, well, they're just ignoring each other. So here's the really crucial part. We take every single one of those meaningful relationships, every friend and enemy pair, and we draw a line connecting them. All of a sudden, our simple list of suspects transforms into this complex, beautiful social network map. It's the entire web of interactions laid out right in front of us. Okay, now that we have this map, our detective can finally zoom out and look for bigger patterns. We're not just looking at pairs anymore. We're hunting for the gangs, the cliques, the teams that all work together. So how do they do that? They use these really smart algorithms called community detection algorithms. And their whole job is to scan that giant network and find these tightly knit clusters. Groups where everyone is highly connected to each other, but they don't have many connections to the rest of the network. And one of the most powerful tools for this has a bit of a mouthful of a name, Weighted Correlation Network Analysis, or WGCNA. You don't need to remember the name, just remember what it does. It's like this master detective that automatically sorts through the entire social network to find the real gangs, the groups that are genuinely working together. And this is the payoff. This heat map is what you get from that WGCNA analysis.
It takes that messy, tangled web of connections we saw before and reorganizes it into these beautiful, clean, color-coded blocks. Each colored square you see here is a different microbial gang or functional team, now totally clear and separated from all the noise. So let's get to the final act. We've gone from raw, chaotic data to a list of suspects, to a social network, and now to these organized gangs. It's time to finally use all of this incredible information to understand the plot of our own health story. And this is where it all comes together. Scientists take these patterns they found, the presence of certain genes or the strength of these microbial gangs, and they feed them into machine learning models. And those models can finally connect the dots between the microbiome's structure and what's happening in the real world, like whether a person is healthy or sick. Now, this isn't just theory. This has real-world, life-saving applications. Take this example from a dangerous E. coli outbreak in Germany. Using these exact techniques, scientists analyzed the genetic data from different samples. And look at this. The analysis perfectly separated the dangerous outbreak strains, those red dots all clustered together, from all the other normal E. coli samples. This gave public health officials a clear genetic fingerprint to track the disease and stop it. But the power of this analysis goes way beyond just fighting outbreaks. It can be used proactively to help us understand and improve our own health on a really personal level. And this is why this should matter to you. In this study, the analysis identified specific microbes that can act as biomarkers, basically signs, for how someone might respond to a certain diet. So if you have microbes from the blue list, a particular diet might work great for you. But if you have microbes from the orange list, maybe not so much. This is opening the door to truly personalized nutrition, getting us away from generic advice and toward tailoring our food choices to our own unique gut. So we started with this impossible book of 1 trillion letters, written in an alien language. But by following this process, finding the characters, mapping their relationships, and identifying the major plot lines, we're finally learning to read it. Our microbiome has so many stories to tell us about our past, our present, and our future health. The only question left is, what are we going to choose to write in the next chapter?